recently, so it used to be resources. So my research is focused on synthetic biology. So here is just a brief introduction. So historically, fossil resources were put into a refinery to get fuels and chemicals, but then starting in the 70s, the first generation of biofuels came about using sugary parts of plants to make ethanol. But going forward, we want to use plant waste or other low-value substrates like fats, oils, and grease to make high-value products, potentially food products like beta-carotene, or fuel products like butanol. And so we do that through fermentation. And fermentation is a pretty complicated process that has to do with the strain genetics and the environment inside of the fermenter. So to balance all that, we can use data science. So I want to talk to you guys about two of my research projects. And the first one has to do with syngas fermentation. So we can take lignocellulosic biomass, that's just a fancy word for non-sugary parts of plants. So we can think about the leaves off the pineapple plants or the pods of cacao. Heat that up with high pressure to convert it into this mixture of gases like hydrogen gas, carbon monoxide, carbon dioxide, and then feed that to this special bacteria, Clostridium carboxylivorans, that can produce ethanol and butanol. So here's the data. We had time course data from 10 different fermentations and 10 different gas compositions. So for the machine learning inputs, we used the current concentration of the 4 different products: ethanol, acetate, butanol, and butyrate, and the gases composition. And we use those to predict the production rates of acetate, ethanol, butyrate, and butanol. And the data was split into training and testing data, so that there wouldn't be showing the machine learning models the answers. So here's some of the data from the trained models. So the blue dots are showing the training data, so that fits pretty well, but that makes sense because it was used to train the model. But then the orange dots are showing the testing data. And each plot is showing one product and one algorithm. So we can summarize that here. And pretty much we see that the random forest and support vector machine algorithms fit the best. And these are not exactly the most complicated models, but for limited training data, we find that using simpler models that are easier to understand is more beneficial. And further, we did feature importance analysis. So we wanted to know if we would want to steer the fermentation towards the more expensive products like butanol, how should we adjust the gas composition? So by increasing the amount of CO2 or CO, like carbon monoxide, you can get more butyrate, and by increasing the amount of hydrogen gas, you can get more butanol. And this was consistent with previous studies, so it's not exactly new information, but it shows how with just limited data, we can learn a lot of biological insights. And we can then apply this for model predictive control. So if we have a live fermentation, we can steer it towards the more expensive products by adjusting the gas. And so this was published, and I always believe in publishing the code as well, so that it can help future researchers with the projects and sort of show your work. So AI and machine learning can be applied well to bioprocess problems, but the issue is that whenever anybody publishes their data, they just sort of attach an Excel file in whatever format they want. So we want to standardize that format. So that's the goal of this project called ImpactDB, or the Industrial Microbiology Publication and AI Crowdsourced Toolbox. It's a live website. You can go to it now, impact-database.com. So here's the interface. So it's organized by species. So this left is showing one yeast species, and then all the different papers from that yeast. And then when you click on one paper, you get this page on the right, which shows the abstract and also the fermentation results. So in this paper, they made citrate. And when you click on an individual fermentation result, you see this information. You can see like the bioprocess conditions. So like what was the volume of the fermentation? What was the substrate that they used? How long did it take? And also the genetics, like which genes were knocked out or overexpressed. And all that is connected to the product titer, which is also known as like the final product concentration. And you might notice this is sort of shaped like a machine learning problem where the features or inputs are on the left and then the targets or like outputs are on the right. So users can upload data, it's crowdsourced, but it's a very laborious process. It can take a grad student 4 hours to extract data from a single paper. So we wanted to use AI to speed up this process. So we call this like AI data extraction. It's a pretty hot field right now. And so it's all human supervised, but you put in different sections of the article into like your AI agent. And then these are the exact prompts that we used. And then you get out data tables that can be used to upload data to ImpactDB. And we wanted to confirm that the AI data extraction was working well. So we compared the results of AI extracted data with human extracted data, and it was 2 different sets of 100 papers, so not the exact same. But we see that the feature variance is very similar, and the silhouette score and the mean difference between clusters is very similar also. So we took this data to mean that the data extraction is working well. And then we trained machine learning models with the extracted data, and they all fit pretty well. This is like a random forest analysis, but the ones that don't work best are like the polyketides, and this is because the data size is small, and these are very like high molecular weight products that don't really fit in with the rest of the data. It's kind of outliers. And again, if you're interested, here's the link to the code and the link to the paper. So ongoing work is developing a large language model interface on the ImpactDB website. And so the idea is you could use ChatGPT, but it doesn't have all the context because the data that it has is sort of disorganized. So using what's called a knowledge graph, you can do retrieval augmented generation, or RAG, and then get more like context-informed outputs. So if you want to know how to make beta carotene, you get like better feedback. The last thing I want to talk about is one of the classes that I teach. It's called like Bioengineering 447, and it is for like teaching Python and then also having students do like hands-on data science and machine learning projects. So the first unit is teaching Python using biological examples. So it's sort of like teaching just the basics like loops and dictionaries and lists, but using things like gene lengths and the central dogma, so converting a string of nucleotides into amino acids. Then the second unit is focused on teaching scikit-learn. This is an open source library for machine learning. And the 2 major functions are model.fit, that's for training data, so for training the model. So you put in like the known inputs and known outputs to train it, and then model.predict, you just put in new inputs and get predictions. And then the third unit is applying those models. So on the left is showing applying a random forest and then doing feature analysis for like a diabetes data set. And then the right is showing like sort of learning curve, so how the loss function goes down as you train a neural network. Our neural networks are different sizes on image data. You can see that like larger neural networks fit the image data better, but they still increase the training time. So here's sort of a breakdown of how the class works. So first, every class grade starts out with some slides to sort of explain the concepts. Then there's a code along period where like, I'm typing a prompt and every all students are following along. And then the last part of the class is the students sort of work independently on a project or like a small exercise. And an important part of the class is the semester-long project, where the students choose a topic that interests them. So it could be their research project or just something that's they care a lot about. And then they need to go through the whole like sort of pipeline and iterations in order to get a validated model, and then they rate a report on their findings. So the last thing I want to talk about is this slide. I show it in every single class. So it's a very hot topic now, like how should generative AI interact in the classroom? And my belief is that it's not going anywhere. Like ChatGPT is only getting better. And we want our students to be very strong in the workforce. So I think that they should be encouraged to use these tools to be like the best versions of themselves possible. But that if they just copy and paste, they won't be learning anything. So there has to be a balance. And I think people have an intuitive sense about like, what's an ethical way to use this and what's not ethical. And I do think that it's pretty clear somebody's just copying and pasting. So it's sort of like they'd be embarrassing themselves. So with that, I want to thank you guys for listening and invite any questions that you might have. Thank you.
Questions for Garrett? We have some.
What is the kind of demographic of students that take your class?
Yeah, so it's a 400 level class, so it's mostly seniors and then some graduate students. And they're almost entirely from either the bioengineering major or like MPBE, like their bioscience and bioengineering. So they don't have much experience with coding. So that's why we need to like teach them Python first. So that does slow down the class, but I think it's good that it's inclusive.
Prereqs?
Prereqs are minimal. It's like one coding class and one like higher level math class.
How do teachers tell like if students are copying and pasting right from? I mean,
it's a good question. So I guess I have a decent amount of experience using it. So there's some quirks like the sentence structure. Sometimes it uses like weird ing verbs or like this like m dash. I guess those are pretty easy to fix. Yeah, that's how I would know.
There are commercial tools that are available to help teachers differentiate between things, basically identify things that can generate by AI.
Do you have graduate students who work on this that might want to partner with a company to develop something that's actually a practical use? We do something that I can see has a lot of overlap with what you're doing.
Yeah. I have a master's student that's entrepreneurial, so potentially could get in contact.
Yeah, I'll talk to you a little bit after this, but yeah, certainly. And then any kind of a granting structure that you've seen where some of this work can probably get supported from a grant.
Yeah. I just submitted an NSF like career proposal for the ImpactDB project, so fingers crossed on that.
Okay.
Nice.
Hey.
All right, thank you everybody.